right, well, good morning. I know some people will be filtering in, but I want to take a moment to welcome Dr. John Mascola. Welcome him to Grand Rounds this morning. So Dr. Mascola is currently the director of the Dale and Betty Bumpers Vaccine Research Center at the National Institute of Allergy and Infectious Diseases at the National Institute of Health in Bethesda, Maryland. In addition, he serves as the chief of the NIAID and NIH Virology Laboratory while holding a concurrent position as an adjunct professor of medicine at the Uniformed Services University of the Health Sciences in the Division of Infectious Diseases at the Walter Reed National Military Medical Center. Throughout his career, Dr. Mascola has been recognized for his dedication to medicine and research. He's received the Meritorious Service Medal from the U.S. Navy for research contributions to the naval medical research. He's received the NIH Director's Award in both 2003 and 2008, as well as receiving the Outstanding Service Medal from the Surgeon General of the U.S. Public Health Service. He has received the NIAID Merit Award on 3 separate occasions for his contributions in research for effective vaccines for human disease, discovery of new HIV-1 monoclonal antibodies, and most recently in 2018 for his contributions to the Smithsonian National Museum of Natural History's Outbreak exhibit. I was furthermore astonished to see that he's also contributed over 350 publications to the literature and peer-reviewed journals, and this is only through chapter 4, I think, of his resume. So there's much more that could be said for Dr. Mascola, but we're certainly privileged to welcome him here to Grand Rounds here at Wake Forest. Let's give him a round of applause. Thank you.
Okay, well, thank you, Joshua, and good morning. And actually, you reminded me about the Outbreak exhibit, which is at the National Museum of Natural History, which is fantastic if you're in D.C. They just did this phenomenal job about the history of infectious diseases going back years and years. They have all kinds of paraphernalia and exhibits, so that's really great. And they just asked me to ask a lot of people to help out with that. But as mentioned, I trained as an infectious disease doc in the military system and then moved over to NIH to do research. I grew up in the era of HIV and became very interested in immunity. And so that's kind of what I'll talk to you about today. Relates to the work we do at my center at NIH, which is vaccine-related work. So I want to start with just a little bit of background because sometimes I get asked, what does it mean to be at the NIH? I talked to the medical students group yesterday, and I think you know this, but it's probably just worth mentioning. So at NIH, we do biomedical research. It's a research institute, and it funds others, academics. The Centers for Disease Control does disease prevention. So if you think disease outbreaks or what flu vaccine to take each year, and the FDA regulates any commercially approved drug or medicine has to go through the FDA. So all of these are sister agencies, part of the Health and Human Services, with somewhat different functions. And then just to mention that the NIH is a really big institute, and it's actually multiple institutes. So I think you recognize those names: National Cancer Institute, National Heart, Lung, and Blood Institute. So my institute is, and Joshua said it right, most people, it's hard to kind of get all the words out, but we're the National Institute of Allergy and Infectious Diseases. And you can see fairly big numbers there. So those funding numbers, if you're interested in research at all in your career, much of the country's medical research is funded by the National Institutes of Health. So you get funding, you write for grants, it usually comes from here. A small amount of that money stays on campus where I work, which is in Bethesda, Maryland, just outside of D.C. This is called the intramural part of NIH. It's actually a small percentage, about 15% of all that money stays on campus. In the center here, if I can get the arrow to work, which I had working, but you can see the main hospital here. And in the back is a center where I work, which is dedicated totally to vaccine research. So I want to briefly tell you what we do, and I'm going to tell you 4 quick vignettes or stories about vaccines. So we were built as a center in 2000, and we were built mainly due to the AIDS HIV pandemic. So at that time, the pandemic was still spreading globally without good evidence that it could be controlled, even though there were antiretroviral drugs. So the idea was to build a center and to do research that was basic research, but also clinical research towards vaccine development. And over the years, we've actually expanded our interest into a lot of different pathogens, mainly viral pathogens, and I'll talk about a little bit about Ebola and RSV, a little bit about flu. And then the last thing I'll mention is that one of the things that we can do that's pretty unique at NIH is we have a research building. We have the ability to make clinical grade drugs, antibodies, and vaccines. So this allows us to do experimental medicine. So we actually have an idea in the lab. We can test it in people, and we do a lot of first-in-human studies. And so the stories I'm going to tell you today are essentially discovery and first-in-human studies and how we think about making a vaccine when you don't have one. So that's the overview. I'm going to give you a little bit about global health, why we need vaccines, very little bit of immunology, 2 slides, I think, and then these 4 vignettes, Ebola, RSV, and universal flu. So just as background, if you look at World Health Organization numbers, infectious diseases are an enormous impact on global health, second leading cause of death overall, and really second to heart disease and stroke, which really affects older population. And the big diseases, you know, TB, HIV, AIDS. If you look at these numbers, malaria, when you see a number like 1 million or 2 million deaths a year, it's hard to wrap your head around that. So for HIV, what does that mean? It means about 4,000 people die a day of HIV in the world. It's not a made-up number, it's a real number. So you can see the impact and the focus on these diseases. If we had better disease prevention, we could save a lot of lives. A lot of these diseases disproportionately affect children, in particular malaria, respiratory disease, diarrheal diseases, respiratory diseases. And then with regard to infectious diseases, there's diseases that emerge that you never knew about before. And that keeps us on our toes, keeps us nervous. Ebola was not known to humanity before it emerged in the 70s. HIV/AIDS likewise. So when I was a medical student, HIV/AIDS was discovered. We didn't know even what it was called at the time. We called it HTLV. Avian flu jumps from birds to people. Everybody worries about a pandemic, SARS, MERS. And you remember Zika from a couple of years ago? It was a pretty much unknown disease discovered in Uganda, few cases, and all of a sudden there were 1 million cases in the northern and southern hemisphere. So just to briefly remind you, so everybody knows smallpox as a vaccine, right? Highly successful vaccine. So what was smallpox? Smallpox, this is 1970s literature that you can find, in a history text. And, they describe it clinically very acutely. So once inhaled, the variola virus invades the oropharyngeal respiratory mucosa, migrates to regional lymph nodes, and begins to multiply. And here's where the problem begins. By day 12, lysis of many infected cells occurs. The virus is found in the bloodstream in large numbers, and a second wave of multiplication occurs in the spleen, bone marrow, and lymph nodes. And the malignant or hemorrhagic form of smallpox occurs about 30% of the time and is often fatal. Terrible, devastating disease. And I think you know the story of Edward Jenner. But it turns out, if you read the history of this, and I went back and looked at it recently, a lot of people made this observation, not just Jenner, that milkmaids didn't get smallpox. But what people figured out, it was their exposure to a closely related virus, cowpox, that gave them immunity. So what Jenner did is he inoculated people with cowpox, which is a related pox virus, and he showed that, in fact, that worked as a vaccine. And over the years, the World Health Organization in the 60s and 70s had this enormous campaign to vaccinate people. If you're older, you still have a little scratch scar. I think you have to be 60 or older to have that, or the 50s. That was your Dryvax vaccine. It was a live replicating vaccine. It caused a little blister, and you got immunity to smallpox. But it was eradicated. We don't need the vaccine anymore. The most successful vaccine I think we have. So if you think of smallpox, and I know you get this question as a physician, why does my child need all these vaccine shots, and is it safe? So you might think we have vaccines for hundreds of diseases, but it turns out we have vaccines for about, and this is throughout all of humanity, all of the world, we have vaccines or licensed vaccines for about 30 infectious diseases. To me, that's not very much, and you know what they are. Your children get DTP, MMR, polio, Hep A, and all that. In this country, we don't get these, but cholera, typhus, rotavirus is an enormously important vaccine for kids. The developing world, where diarrhea and dehydration causes severe morbidity and even death. Yellow fever keeps getting worse with global warming, but we have vaccines for that. And so the point I want to make, because I'm going to talk about modern vaccines, is that this vaccine science is not new. It's enormously effective, but the first vaccine, DTP, actually came out in about the 20s. So this is pretty old, effective technology for these vaccines. And how do we do it? So I'm more of a virologist. I went into the world of HIV, and I learned, didn't get a PhD, I just learned HIV at the bench and spent years in the lab. So I'll focus more on viruses, but you can take a virus and make it live attenuated. So that's polio and MMR. They are live viruses passed in tissue culture until they're attenuated. So you get infected, but it's such a mild case that you're not sick and you get immunity. You can kill the virus with a chemical like formaldehyde. That works. That's influenza, hepatitis A, and the more common polio. So the old polio was OPV, oral polio vaccine. In this country, we've switched to IPV, which is inactivated polio. So there's just no risk of a polio infection. In the 80s, we learned how to make proteins in the laboratory, recombinant proteins. Hepatitis B is the first recombinant protein vaccine. And you hear about the HPV vaccine, even commercials on TV. That's a cool technology of a virus particle with no genes inside it. And that's also an enormously effective vaccine. So those are more modern approaches. All of these approaches try to do the same thing. You induce the immune system to make antibodies and T cells. Antibodies block the virus. T cells, especially CD8 cells, clear any virally infected cells. So if you can block infection, even if you get a little bit of virus in the system, your CD8 T cells kill it, but your immune system remembers seeing it and you're immune. So that's the basic immunology we try to do. So the vaccines we know of are enormously effective. This is CDC slides. You can find these on their website, and it's a log scale. So peak your disease, the rate of that disease compared to when it was at its peak has gone down 100-fold, 1,000-fold, or is essentially gone. Polio, smallpox, diphtheria, we don't see in this country. You see them elsewhere globally, but not in this country. But I wanted to just highlight for you a couple of diseases, measles, mumps, rubella. So I would guess no one has seen these. Maybe pediatricians have seen a case of mumps occasionally, but these are diseases you don't see, even infectious diseases docs don't see. But if you look at measles, so when I was a kid, in the early 1960s, there were 760,000 cases of measles. I remember getting mumps, but the vaccine came shortly after that, and these diseases essentially went away. And the disease I'll highlight is congenital rubella syndrome, which is also no longer found in this country. Probably no one has seen it here. A devastating disease of young kids. And in a 3-year period in the early 1960s, there were 12 million rubella cases in this country, and which means that pregnant women occasionally got rubella, not occasionally. 2,000 cases of babies born with encephalitis, 20,000 children born with congenital rubella syndrome. You see the classic triad: congenital heart disease, often PDA, but others, ocular defects, which can lead to blindness due to retinal pathology, and hearing loss. So a pretty devastating congenital abnormality gone from this country due to vaccines. So that's all success, and so there's not much to talk to you about, except there are some diseases where we wish we had vaccines. So hepatitis C, norovirus, which is also Norwalk virus, caused on cruise ships, diarrheal diseases, enterovirus, cold virus, HIV, RSV. CMV is interesting because CMV causes congenital abnormalities, and it's the most common cause of neonatal congenital abnormalities now that rubella is gone, and we don't have a vaccine for CMV. So in 2014, you remember there was an Ebola outbreak in Western Africa, and there were a couple of cases that came to the United States. It was kind of a big deal. There was a worry that somebody might walk off an airplane in New York. And so CNN had a story, and the gist of the story was, in the middle of the Ebola outbreak, what's the scariest virus in the U.S.? And their point was, it's not Ebola, it's flu, or it's norovirus, or hepatitis C, or enterovirus. And they were right. And so when we think of, so I'm a vaccinologist now, and when we think of where's the opportunity, where do we need to do research, where do we need to do discovery? TB, malaria, HIV, the ones I mentioned before. But if I could ask, are there any folks in the room that are pediatric trained at all? Is it all medicine? It's like 99% medicine. That's okay. So one person. But you should know RSV is respiratory syncytial virus. And the ID folks see it in the elderly. Dr. Sanders was telling me that. But it's a major problem in children. So what happens when we don't have a vaccine? Right? So we have all these great vaccines. What happens when we don't? So here's what we know. On the antibody side, viruses have a surface protein. That surface protein is how they attach to a cell and get in. So if you bind to that surface protein, oftentimes you have a very good neutralizing antibody, and that's a key component of a vaccine. Most vaccines are measured by how good their antibody response is. The hepatitis B vaccine is essentially the surface antigen. Everybody knows hepatitis B surface antigen. You measure it in the blood. That's the vaccine. You make it in yeast and formulate it, and it's very immunogenic, and it's an amazingly effective vaccine. When you get immunized with influenza, the main antibody response you're looking for is to the surface protein called hemagglutinin. So a quick story about HIV, which I grew up with. The field thought that this spike protein, the infectious disease docs will know it's GP160. If you make an antibody response to GP160, you'll have a vaccine, just like hepatitis B. Should work fine. So here's some nice pictures. I work with structural biologists, so when you see nice pictures, I've borrowed them all from people who do X-ray crystallography. But so viruses have spike proteins. That's how they get into cells. And if you turn it around, here's the HIV. It binds to CD4 T cells. I think you remember that. Then the protein rearranges. It sticks out a piece of itself, binds to CCR5, and then gets into the cell. So we know how HIV works, right? So we know a lot about it. We know its immunopathogenesis and all that. So here was the vaccine that we tried. Take the spike protein, make it in the lab as a recombinant protein, immunize people. It turns out it's what we call GP120, which was a part of the 160. And again, ID folks will recognize those terms, make antibodies. And this was done in the early 2000s in a big trial. See if it works. It was published a few years later, and it was published in Journal of Infectious Disease, and the conclusion was there was no overall protective effect on acquisition of HIV infection and no effect on viral load. Well-conducted trial in high-risk people. Kaplan-Meier curves overlapped. Very well-conducted study. It was essentially as if you were giving placebo to people. It didn't work at all. So this was stunning to the field, caused a lot of retrospective analysis. So I studied that problem because when I went into the lab, I had a great mentor, and he suggested I kind of try to understand what's going on with antibody responses. So it turns out that when you immunize, you get a lot of antibodies to HIV. But if you study HIV that really buds from a cell, and the way you grow HIV in the laboratory is you take cells from an infected person, isolate their peripheral blood mononuclear cells, stimulate them so the virus comes out, and then you get some healthy donor PBMCs, and the virus goes into those and grows. So you can get a lot of virus, you can study it. It turns out actual HIV, when you grow it, you can see that it's very highly sugared or glycosylated. That's its natural state. It buds from a cell, and it buds, that viral spike has a lot of sugar on it. That sugar or carbohydrate protects it from the antibody response. And that is an evolutionary trick that viruses use, particularly chronic viruses. If HIV is going to replicate in the blood for years, it can't be eradicated by your antibody response or it wouldn't exist. So evolutionarily, it has figured out tricks to do that. And over years, I'm just using visuals here, but there's a lot of biochemical studies by a lot of people. What we learned is that here's all the sugars of the glycans, but antibodies occasionally find a vulnerable spot in the virus and get in and neutralize. It's just that our vaccine didn't make the right antibodies. So we had to go back and figure out how the immune system does this for HIV. So I did some studies early in my career after I left finished ID fellowship and went into the lab. So if you look at HIV, this is what we knew, and we still know is an early burst of viremia. And antibodies come. It takes the immune system a while to make neutralizing antibodies. But if you study people and find occasional people who are slow progressors, the immune system is intact, they eventually make really good antibody responses. It's too late for them to eradicate the virus, but those antibodies are interesting. So you can isolate them from their blood, and the way you do that is a technology that emerged in the early 2000s, which is common now. So I studied with some colleagues about 100 people who were HIV infected before they started antiretroviral drugs, because in those days we didn't start till their CD4 count started to drop. And some of those people had these really good serum antibody responses. So we said, what do those antibodies look like? How can it help us make a vaccine? The technology that I think you should know about, because you hear a lot about licensed antibodies, is now used all the time. It used to be tricky technologically, now it's easy. You take a person's B cell from their blood, you can isolate B cells pretty easily. You can incubate them with a fluorochrome protein, do basic flow cytometry and say, hey, these are the B cells reacting with HIV protein. From a single B cell, you can do PCR and recover the antibody genes, and you can express them in the lab. So in a matter of a couple weeks, you can have any antibody you want from any B cell you want, and so you can find really good antibodies. And this has changed the world of vaccinology. It's changed the world of cancer therapeutics to get these antibodies. So we did that, and at that center that I mentioned, I work with people who are formally trained structural biologists. If you have a protein, a sugared protein, it's hard to get a crystal structure, but if you bind it to an antibody, oftentimes the protein will crystallize. If you can get it to crystallize, you can do X-ray crystallography. So this photo is not just a nice picture. This is an atomic level resolution of what HIV looks like. Here's GP120, there's 3 of them. Here's GP41, here's the antibody binding. And you can see this atom to atom. You can pull it up on your computer. You can rotate it. You can look at it in 3 dimensions. You can find whether it's a cysteine, whether it's a serine, whatever you want. So this kind of information has also changed what we do. And so I'll show you a quick movie here. So these antibodies are drawn to scale. So this is about what an antibody looks like in real scale compared to the viral spike of an antibody. So antibodies are big molecules. If they bind a virus, it's not hard to figure out why they interrupt the entry process. They basically, we study this in detail, how do they do it, but they basically muck up the entry process and the virus can't get in, and that's a good thing. So what we've tried to do with HIV, and I'll tell you right off the bat that we haven't figured out HIV yet, and the 3 other stories I'll tell you we have more success, is to take these antibodies and say, okay, I know where it binds, so let me make a vaccine by the reverse thinking. I'm going to define where the antibody binds, and I'm going to make that part of the virus as my vaccine. And that's what that says. And so you can do that. You define your epitope. So we call this structure-based vaccine design. And so the world of vaccinology has moved in this direction. You take your sub-epitope, you use it as your vaccine, and maybe you boost the response with something that looks a bit more natural, and you try to teach the immune system to pick a spot on the virus. So we've successfully done that in animals. It's being tested in people. But frankly, a vaccine for HIV is years away, 5-10 years away, because the process of bringing this through clinical development is slow. But there's an alternative that's faster. And what if we could just give the antibodies to people? So this antibody that works so well, it totally blocks viral entry in the laboratory. It works beautifully. So what if we could give it to people directly? So that's akin to using antiretroviral drugs for PrEP. You've heard the term PrEP. Somebody takes a pill every day like Truvada, and it can actually block them or prevent them from getting infected. Antibodies are pretty cool because if you give an antibody to someone, it can last in the system for weeks or even months. So there's a potential advantage if it could work for infectious disease. 1 shot of an antibody could protect someone for 3 months or 6 months if we could show it could work. So again, we're very fortunate in our center. We actually can make what's called an investigational drug product. We could make the antibody. So we actually isolate it from a person. We made it. It's not, I'll emphasize, a licensed drug yet because we've never put it in a person yet. But we do the experimental trial, and to do that, you go to the FDA, you file an investigational new drug application. You say, I've made the drug product by all the appropriate processes, GMP, and I want to test it in people. And we do that. And if you give an antibody to healthy volunteers, you give it, and it has a half-life of about 3 weeks. And this is known for years and years. You see commercials for therapeutic antibodies in cancer. So antibodies last, if the half-life's at 3 weeks, they may last a few months in the blood. That's a good thing. But it turns out there's technologies to make antibodies last longer in the blood. I won't get into all the details, but antibodies bind to a receptor called the neonatal FC receptor. If you alter that affinity, you can get the cell to recycle antibody, kick it back out into the blood. So we did that. We added this mutation, which we didn't discover, but we were the first to test it in people. And this is 1 infusion of an antibody, and here's 6 months of follow-up. So the antibody lasts a long time. And this is an HIV antibody in a healthy donor. So this is just proof of concept. What we think is possible is that this could protect a person. But we have to test it, and testing in vaccines is expensive and difficult because you have to do large placebo-controlled trials. Here's why we think this could work. So what we do know about HIV is that when someone is exposed, this vaginal mucosa, foreskin mucosa, I'm not a very good artist, there's a lot of virus, but if you look at what infects a person, it's phylogenetically a monotypic virus. It's 1. So only 1 is able to compete out and get infect. So we think antibodies could block that event. So that's the hope. And the trial is basically, so it's an international placebo-controlled trial. It probably cost $10, $20, $30 million to conduct a trial like this. So this is a large international multi-center cohort. And it's being done in people who are at high risk, who have a history of sexually transmitted diseases or exposures. And those groups are either high-risk men in the Americas, high-risk women in certain regions of Southern Africa, where their community cohort is a prevalence of 30% for HIV. So there's no way for them to avoid HIV in their cohort. Then you don't even know who's infected. So the study is placebo-controlled. because that's the only way to know if it would work, and it's ongoing. So here are all the sites, and basically this cohort of people is fully enrolled. They're getting the antibody once every 2 months, and we'll have an answer probably within 6 months to tell us if an antibody can prevent HIV infection. So it's a little bit sort of discouraging it takes so long to get answers. When you do therapeutic work, you get answers more quickly. So this is a brief story of an Ebola antibody that came out of some work we did where we could show literally in a year that it works. So I think you all know about Ebola, fatal hemorrhagic fever disease, emerged in the 1960s. There's an ongoing outbreak right now. I don't know if people are very much aware of that, but in the Democratic Republic of Congo, there's an outbreak. Average mortality is about 60%. You know, you see it on TV. It doesn't quite. A number of my colleagues have gone to DRC, taking care of patients. It is almost as bad as you might think. Hemorrhagic fever in its late stages is a terrible disease. It also turns out to be more communicable than we thought. So the protective gear that one has to wear is really quite intense because there were nurses getting infected and doctors getting infected because they weren't careful enough. There's no vaccine or treatment for Ebola. So a few years ago, we isolated an antibody. It was an interesting story. A physician in the DRC became infected in the 1990s. He worked with us. He survived that infection. He continued to take care of people in other outbreaks, and his immune system was very revved up to Ebola, probably because he was exposed several times. So he came to NIH and let us draw his blood and let us isolate that antibody just the way I showed you, same technology. And we chose a B cell that had a really good antibody. So we had this fantastic antibody against Ebola. We meaning the structural biologist, solved its structure, so we knew how it worked. It made sense why it blocked. And again, we were able to make it as an experimental product. And it's an interesting story because we're going to do the same thing. You have to go slowly when you're testing a new drug or a new antibody in people. You give it to healthy people in safety. As we were about to put it in people, just to make sure, and it always is safe, it's an antibody, it's a protein, but the outbreak, the current 2018 outbreak, emerged. So we received a letter formally to the head of the Institute from the Ministry of Health in the DRC saying, we know you have this antibody, we know it's in people. We are asking if we could use it as an investigational agent because people are dying of Ebola. There is a process for that in this country. The FDA has a process called an emergency use authorization. You may engage that in your own practice someday. If there is a drug being tested, could be a cancer therapeutic, could be an antibiotic. It is not licensed yet, but a clinical trial shows it works, and they're in the process of getting it commercially licensed. You can get that drug as an emergency use authorization. It takes some paperwork. But we did that, emergency use authorization, and it turns out we were not the only one to have a drug. There were 3 different antibodies from 3 different groups, which is a good thing, and 1 small molecule drug. So what investigators in the Democratic DRC did is they said, we're going to test all 4. No placebo, because you can't, all of these drugs worked in the lab, and they worked in monkeys. So we're not going to give a placebo. We're going to give the 4 drugs head-to-head in a randomized trial, do the paperwork, do the randomization, and it turns out that 2 of the drugs worked and 2 didn't. And it shows you why you have to do human clinical trials. In the lab, all looked good. In monkeys, which it turns out monkeys get Ebola, all of them worked. But one of the drugs that worked was the one we isolated. The sickest patients in this study had a mortality of 55%, and these were people that were dying. They had organ failure, and that mortality could be brought down to 30%. People who came in early, you could bring the mortality down to 10%. It's the first time the world has ever had anything that effectively treats Ebola, and it really comes from the vaccine technologies of pulling out antibodies. So I'm going to tell you 2 other success stories from the world of vaccinology. Success in the sense that we think we have vaccines that will work, but they're not on the market yet. So RSV, because you're internists, I'll tell you that it causes severe pneumonia in children. I think you all know that. But what you may not know, it's the leading cause of hospitalization in this country in children less than a year of age. Pediatricians know that. Worldwide, it's a major problem, and the world has been working on vaccines for many years. From the world of big pharma, Merck and GSK, this is the vaccine they want because in this country, if they had the vaccine, every child would get it, like DTP, like MMR, because it's a major problem. So in the 1960s, a vaccine was tested, and it didn't work. We actually found a vial of the vaccine in a freezer at NIH, and looked at it, so what it looked like in those days. The vaccine elicited a pretty weak response. When it was formally tested in children in the Washington, D.C. area in a placebo-controlled trial, it did not work. It did not prevent RSV. And in fact, some of the children got an enhanced respiratory disease, which was kind of an enhanced pneumonitis, so that was kind of hard to understand. The result of that was for the ensuing 50 years, there has been no RSV vaccine, and people were actually afraid to try again because the vaccine seemed to cause harm. So this is just a classic example where you really need to go back to some scientific understanding and then restart the process. That scientific understanding came from structural biologists, and this is, to me, one of the most gratifying parts of being where I am at NIH. I lead that center. I'm an MD. We're very fortunate to have PhD structural biologists. Their lab is right next to a lab of a virologist named Barney Graham, who studied RSV for 20 years, one of the country's experts in RSV. And it's actually a true story that Barney was studying RSV. This is the spike protein. And a fellow in the structural biology group went over to him and said, I'd like to solve the structure of the spike protein. It's never been done. So in 2013, that fellow solved the crystal structure, just the way I told you, and he found there were two forms of the protein. There was the form you expected, which was the normal-looking fusion protein that works, gets into cells, and there was this unusually-looking post-fusion protein, which is what the virus looks like after it's entered the cell. But in culture, both of these exist. And it turned out, Barney worked with the structural biologists to figure this out. If you look what was in the vial, all of the protein was essentially triggered into the post-fusion state. It was the inactive form. So what happened when they chemically inactivated this virus, they left it on the bench for 24 hours with formaldehyde, and all the protein essentially became triggered or mucked up, for lack of a better word. So we were giving a vaccine in this case which had no active protein, at least no protein in the right form. So we know why it didn't work. And we know what to do about it, potentially. Keep the protein in its native form. That's the form that enters the cell. If the immune system sees the native form, it's going to make an antibody response. The structural biologists basically figured out how to make a couple of simple mutations. There's 2 amino acids here, and this is really how simple, this is why an MD like me can understand structural biology. There's 2 amino acids here that are close by. When the protein triggers and changes form, they're far apart, because the protein actually extends itself to enter the cell. If you put a cysteine bond here, you keep these 2 amino acids together, and in fact, the protein now can't trigger, can't change its form. They made a couple other mutations, and we could make a protein in the lab. Looks like this, which is basically a very nice, well-formed RSV surface protein, and it's stable. If you make that and put it in people, you get this really robust immune response. So people have tried several iterations of RSV vaccines. You never could boost the immune response. So remember, we're all exposed to RSV as kids. So you have some response, but it's waned in adulthood. So when we give this vaccine to people, you get this really high boost in response. So now you have a vaccine which you could go back, potentially, and give either to women who are pregnant, induce their immune response or heighten it, and those antibodies get transferred, and when the baby's born, particularly if the babies are premature, they're protected or would be protected. So this vaccine is not on the market yet, but it's been licensed for development. The process of licensing means that the pharmaceutical company actually has to prove formally it works. That's a 4 or 5 year process. They actually have to do a placebo-controlled trial to show it works. The thinking is that it's likely it will work, but you never know until you test it. So the last story is on influenza. I think everybody knows influenza well. It's not such a bad disease most of the time, but in fact, it really is. So, there are millions of cases a year. That's not surprising. But what does that mean? Globally, there's probably around 500,000 deaths a year from influenza. In this country, most of the deaths tend to occur in the older population, but there are a lot more influenza deaths than you would think, and you can find this by just looking on the CDC website. So I just pulled numbers from the CDC website. You can, you Google it, it comes up right away. 15 to 45,000 people die a year from influenza. To me, it's a pretty shocking number. The reason it's not news is it happens every year. We're just used to it. Last year was the worst year in decades for influenza. 80,000 people died of influenza. So we need a much better vaccine. We have a vaccine. It tends to be cumbersome and doesn't work all that well. So It's an old technology. We grow it in eggs because influenza was first an avian virus, jumped over to people. It likes to grow in chicken eggs. You grow it. This is a very cumbersome process. The people at Sanofi told us that they ship in something like 500,000 eggs a week over the course of a few months to make the virus, and this is every year. So this is pretty old technology. Those of us who are into modern vaccinology would like to modernize this. So you might remember that there's 4 strains of influenza. The ones that get you that are bad are H1 and H3. They cause the severe pneumonia that will even make a young person sick, put you in bed for a week. So should you get the vaccine? Yes, because the vaccine, although the effectiveness overall is between sometimes 20 and 60%, it does prevent severe influenza, severe pneumonia. I just got the vaccine, you should get it, but it's really not what it should be. So problems with the vaccine, we get it every year, right? That's not a good vaccine. MMR, the last time you got your MMR was just before you went to college, and you're good for the rest of your life. We changed the vaccine, so we change the strain of it every year. MMR's never changed. Polio doesn't change. You give the same vaccine. The flu we change almost every year. So only last year we change it. So what can we do about it? What we want is obvious. We want it to be like all the other vaccines we have. We want it to be 90% effective, to work so we don't have to change it, and for it to last, let's say, 10 years. It would be great if you had your flu vaccine and it lasts 10 years. So if you look at flu, so we go back to the science again and say, what can we do? If you look at the hemagglutinin protein and make a phylogenetic tree just based on its sequence, here's H1 that circulates, here's H3 that circulates. They're pretty different, so you can see why you need both. The strains are pretty different, and these circulate each year. And then we worry about avian influenza jumping to people. So H5 jumped over to people and killed hundreds of people in the Southeast Asian region. Fortunately, it didn't spread human to human very well. But when avian flu jumps to people, H7 jumped, it can be fatal. And there's this major concern that it only takes a few mutations for H5 to become comfortable replicating in people. And if it becomes comfortable replicating in people, you have an avian flu epidemic. And there's a lot of concern, and that is not far-fetched. So there's a lot of interest, and we are funded along with a lot of other people. There's actually been a major investment. So from our perspective, we do what we did for RSV, we do what we did for HIV. We look at the protein, and it turns out, so viruses have been around for thousands of years. They exist because they have evolutionarily figured out how to exist. Flu exists because the hemagglutinin changes to evade the immune system. Most of the antibody response to flu is to these red variable regions. So here, again, you just go into your software program with a structure. You don't have to be a structural biologist. And you say, show me the surface structure, color the amino acids according to variability of influenza. And the red is variable, white is not variable. So when you get immunized, the reason you get immunized and change every year is your immune response to this year's vaccine won't cover the virus if it changes a little bit next year. So flu is tricky. But there are conserved areas of flu. So one of the major areas of flu vaccinology that's in the clinic, it's in phase 1 testing, is to teach the immune system to target. Every virus has vulnerable regions. HIV has vulnerable regions. RSV was pretty easy when you stabilized it. Flu has vulnerable regions. So what we work with the structural biologists to do is we want to focus all the immune response here. So we made what we call a headless HA. You can basically just chop off the head. If you chop off the head of a protein falls apart. So you need very savvy structural biologists to put some linkers in here, put on a motif down here, and the protein now stays together beautifully. And to make it a bit more stable and immunogenic, you can put it on a nanoparticle like this. So this is the stem of hemagglutinin. This is what it looks like under a cryo-EM, under a transmission EM. So this vaccine is in people. And the question is, and it's at that stage of phase 1 testing, can you induce immune response to the stem, and will that be protective? The other approach that we and others are doing is there are also technologies now where you can put a whole bunch of different proteins on one particle. So you could take all of the circulating influenza strains, that's H1 and H3, and there's 2 Bs in the virus. So if you get your vaccine, it's 4 different strains. You can put them all in one particle. You can take the avian strains that we're worried about jumping to people. You can put them all in a particle, and you could have broad immunity in one vaccine. So these technologies take some time. They have to go through testing and all of that. The last technology I'll tell you about, is a very cool technology that you will hear a lot about. It's being used in cancer immunotherapy a lot. It's beginning to be used in vaccines, and that's genetic immunization. Genetic immunization means that instead of using the hemagglutinin as a protein, I'm just going to take the gene. I'm going to put the gene in the DNA plasmid, which is really easy to do. That's basic laboratory recombinant protein expression. And it turns out if you make the plasmid the right way with a promoter and some good molecular biology and inject it into a muscle, the plasmid is taken up by muscle cells, and the protein is actually translated. You get transcription and translation. The muscle cells pump out a bunch of hemagglutinin. So you're basically given a vaccine without any protein. That is an enormously easy technology to use. It's being done now actually with RNA, which works better. When you make RNA in the lab, it's a little unstable, so you have to put it in a lipid nanoparticle, and people have figured out some tricks. But RNA works well because you do the same thing. You inject it, and so it's not DNA. And the fact it gets into the cytoplasm and it gets translated right away. It makes protein right away. So it's very efficient. You get a bunch of cells to make whatever protein you want. You can make Ebola this way. You can make RSV this way. You can make flu this way. So there's a whole new world of vaccinology that I think in the next 5 years you will see vaccines that are basically DNA or RNA vaccines just because they're so facile. So for flu, what does that mean? If flu changes, or if you get avian flu, all we need, the CDC will go on site, they'll sequence the virus, they'll send us the sequence digitally, and we make this RNA vaccine. That all happens in a matter of a few weeks. Then you have to work with an industry partner to make the vaccine. That only takes a couple months. You can have vaccines very rapidly. So this is just a summary of everything I told you. I hope it's not too scientifically oriented. There's certainly no specific translational aspects to what you can do for your patients today, but I think it will help you understand what's coming. All of these technologies for viruses rely on understanding, how viruses work, and the same thing applies to bacterial toxins, staph toxin, vaccinology, and all of that. Nanoparticles will be in the clinic. They are for cancer. Genetic immunization will be in the clinic. They are already for cancer. And I just want to again acknowledge, so as I mentioned, I work at NIH. There are MDs and PhDs that work together in this center. There are people who are experts in, just to acknowledge them, respiratory syncytial virus, Byron Graham, Ebola virus, Nancy Sullivan, structural biology, Peter Kwong, anything to do with clinical trials, Julie Ledgerwood. So I'm very privileged to work with all those people, and the way you get this kind of work done, biomedical research done nowadays, is collaboratively. And so if you have any interest in that, I would encourage you to find really good collaborators who are expert in whatever you want to work in, and you can get a lot of good research done. So thanks for the opportunity to talk.
Great. Well, thank you for that talk. At this time, we open it up for questions. And if you could, there's some feedback forms that are in your chairs. Take some time to fill out those feedback forms. They were also emailed out. Is there anybody that has a question? Doctor.
Thanks. That was really terrific. Had a really good experience there. Thank you. Question is, I think if I remember those original HIV vaccine trials, there was concern and I think maybe even a small signal that some antibodies might increase infection risk. So by mutating that FC portion, do you mitigate the fact that it might be taken up by cells that could be infected? And is that a strategy besides extending the half-life that might decrease that risk?
There was a trial that showed a signal for enhanced infection. But in fact, it was the one vaccine that didn't induce antibody. It was a Merck product that had internal proteins that induced CD8 response. Maybe it's a camp. So that enhancement was not to be antibody-mediated, like dengue, where it's not to be antibody-mediated. So that mutation in the FC, we don't think will mitigate antibody enhancement or actually play a role in that. It's solely for the purposes of the half-life in that case.
Thanks. This has been a marvelous review and preview, hopefully. I appreciate it. Everybody here knows I'm going to ask the aging question. So, and especially Zoster and the things that older folks worry a lot about. What's the future? What would be your strategy?
Yeah, so, I think the Shingrix vaccine is a great example of the future. So if you see the commercials, maybe you ignore them for Shingrix. Zoster Vaccines, so it's the chickenpox virus, and in the days when chickenpox was common, when you got it as a child, your immunity was really good, although occasionally you get reactivation, you get shingles. And it turns out that if you revaccinate, you can prevent shingles. But the original vaccine didn't work well in older people, and so its efficacy wasn't very good. The current vaccine works really well. It's from GlaxoSmithKline, and it works really well because the adjuvant is very reactogenic. It actually causes quite an indurated spot, but that means that it induces a very strong CD8 T-cell response, which is what holds the varicella virus in check. So you can overcome the somewhat attenuated immune response even in older populations with very strong adjuvants. Nanoparticles can do that, adjuvants can do that, so I think that's part of the future.
Thank you. Oh, maybe this will predict. The AI question. So Google AI has been shown to predict gender now from retinal patterns, AUC up to 0.97, and they're getting better and better than, say, radiologists. So machine learning, maybe patterns for vulnerability, heat maps for these viruses, is that going to inform your future?
Yes, absolutely. And, it's not specifically my area, but there are huge efforts, some funded by the U.S. government called PREDICT, which is trying to predict which viruses come up. Some recently by the, I can't remember if it's the Page Family Foundation or the Gates Foundation, but the philanthropic groups, because they come from this technical background, have this huge interest in funding machine learning algorithms to help predict viral outbreaks. And so, that's a major effort. How good it will be, I think, remains to be seen. It certainly is being used to predict influenza with some success. It's being used to look when dengue will reemerge and where it's going to go, yellow fever, that kind of thing.
How about for the crystallography, the graphical? It seems like it's graphical. Is it as much graphical as it is?
Yeah, the crystallography is pretty chemical, and it's been around for years, so it's basically atomic resolution, where that used to be hard to do and very much a nuanced field to structural biologists. And really what's changed is that you can download software on your own computer and pull up a crystal that someone has solved and look at it yourself. So it's just become more accessible. But, with regard to whether there's machine learning on that, that's, I think, a harder area.
I speak loud, so
Yeah.
I'm a medical scholar, and immunotherapy has basically revolutionized our field.
And we learn that, the cancer inhibits the immune system, and one way to overcome that is to find the signal from the cancer cells that promotes that inhibition. So we have PD-1 inhibitors and CTLA-4 inhibitors that basically revolutionized the treatment of melanoma, lung cancer. Now we have 5-year survival with metastatic disease. So you focus a lot on how to activate the immune system by using, a passive immunotherapy with immunoglobulins and vaccines. What about finding the signal that HIV promotes to inhibit the immune system? I know that is, an infection with the CD4 cells or that, induces the destruction of the cells and so forth. But do you have a signal that you can inhibit and brings the immune system back?
Yes, so I think there's two parts to that question. And what I would say is the infectious disease world is learning from the oncology field, in part because you can do more in the oncology field with sick patients, and oftentimes in the vaccine field we're dealing with healthy patients. But for HIV in particular, the answer is yes. People are trying to look for specific inhibitory signals that the virus uses and interrupt those as part of immune therapies. And there's a number of nuances that, first of all, you can try latency reversal. So could you actually cure HIV infection? So we suppress it very effectively with drugs. But as soon as those drugs come back, viruses already 10 to the 5th, 10 to the 6th in 2 weeks. Could you actually reverse latency under the auspices of antibody or drugs, kick out the virus and then start to kill all the reservoir in the body. That's one approach. And then in my world in vaccines, what we're trying to learn from checkpoint inhibitors is how could you take advantage of those checkpoint release signals? So when you vaccinate, you get a more revved up vaccine response. You know, we can't give PD-1 to healthy people necessarily, but we can understand that pathway and try to use an adjuvant to engage that. So I think broadly speaking, there's a lot of learning from oncology to infectious disease and vaccines.
All right, well, thank you all.
Thank you.